continue with the ceremony. So, as you know, the Institut Pasteur is leading a network, an international network with more than 30 different institutes around the world. And we have the pleasure to have here a few representatives of those institutes. And so they will be introduced by Pierre-Marie Girard, Vice President for International Affairs of the Institut Pasteur.
Thank you. It's really a great pleasure to be there, and an honor and pleasure to welcome all these young scientists coming from all around the world. I should say also that if we look at the origins of the students here in Pasteur Institute, we know that we have more than 4 continents who are represented, with 19 countries each year. And that more than 300 students out of France are permanently on the campus of the Institut Pasteur of Paris. So since more than 120 years, scientists from Institut Pasteur are committed worldwide to improve public health and to contribute to research. With and these groups of Institut Pasteur are now included in the network that we call the Institut Pasteur, International Pasteur Institute network. And it's a tradition for this ceremony today that we always have representatives from fellows, scientists, PhD, young PhD, coming from other institutes than Paris one from the network. Of course, Paris Institut Pasteur is one of the 32 institutes of the network. The Institut Pasteur in Paris attributes several grants, and particularly fellowships for students, particularly for PhDs, and thanks to what we call the Calmette and Yersin program. We can support not a large number, but around 4 doctoral grants each year. And we also give some facilities for short stays, mobilities for doctorants. Today, we have 4 young PhD coming all from Europe, which is not always the case. And but European is, of course, one of the parts, an important part of the 5 continents who are committed in our network. And also, I'm very happy to note that all the 4 representatives today are young ladies. So congratulations for that. And I would like to quote these 4 people who are seated there. Annabelle Perrier, can raise the hand. Just in a few words, she is coming from the Université de Lille. Université de Lille is one of the 4 institutes that we have in France: Paris, Lille, Guadeloupe in West Indies, and Guyane, which those 2 are departments in France. So we are very happy, and she worked on the trafficking of the M protein, on the Middle East respiratory syndrome coronavirus, and helped to understand how to contribute to the better knowledge of the viral cycle of coronaviruses. Thank you. Then Régine Kiazoua Mbengue, coming from Brussels. The institute is called Scienzano. It's very interesting. She is not in biology, but she is in public health. And she worked on the evolution of the socioeconomic position after cancer of Belgian workers, measure and determinants. That means that she worked on a huge database to try to understand the evolution of the socioeconomic position of Belgian workers who had cancer. Thank you. Then we have Irini Papazian, coming from Greece, from the Hellenic Pasteur Institute. She worked on the roles of soluble and transmembrane TNF in the central nervous system, and she contributed to explore how we can maybe fight against the neurological side effects on CNS of the TNF drugs. And so she more precisely studied the interface between the immune system and the CNS. Thank you very much. Then we have Adelina Stoyanova, coming from the Stefan Angelov Institute of Microbiology in Bulgaria. She worked on the of new strategies for treating in vitro, I guess, Coxsackie virus B infections with a quite original concept, which is not to give drugs together, if I understood well, but to give them sequentially. That's correct? So all the four did wonderful works, and as the director of the International Affairs here, I'm really very pleased to have you today. We know that we couldn't invite all the scientists, young scientists like you, who had their PhD this year, but you are very symbolic of this young generation from outside of Paris. Thank you very much.
So we'll start now with the graduation ceremony. The first PhD will be Regine Kiazua Benji, that she will present her work. And she will receive the diploma from Monica Sala and Pierre-Marie Girard.
Good afternoon, everybody. So I'm Regine Kiassois from Cienzano, which is the National Institute of Public Health in Belgium, and I will present you the main achievement I did in my study about the socioeconomic position evolution of the cancer workers. So I started to study this topic because of three main realities. First, the difficulty for workers with cancer to manage work, but also the unfair economic burden on the family, and also that could lead to social exclusion, and also the stigma that are still around cancer and people who do work with cancer. In order to investigate this topic, we conducted 2 literature reviews, 3 cohort studies, and 1 observational study surveying cancer patients. And for this, the third cohort study, it was a population-based study for which I coupled data from different registers in Belgium. It was a nightmare because of ethical, legal, and technical reason. So I quite often look like this during these 4 years. But finally, we got the data more or less on time to just get some important results. So first, the need to be assessed as soon as possible, and especially the need of the workers. The fact that the young workers, especially those who are the less educated, are really at risk to be unemployed, which is really unfair and something that we need to avoid. Also, the nature of the determinant of the unemployment among cancer workers vary across the time. The fact that the return to work need to be integrated in the care pathway as soon as possible and not be managed aside. But also the fact that the system, especially the social security system, need to work not only on economic incentive, but also supporting rehabilitation. Thank you.
So next PhD will be Irini Papazian from Greece.
Good afternoon, everyone. I did my PhD at Hellenic Pasteur Institute in Greece at Molecular Genetic Lab. So we know that TNF inhibitors are blockbuster drugs for many inflammatory diseases, but why do they make multiple sclerosis worse? The only protective effect that we know is that TNF protects neurons. So in order to investigate this, we made some sophisticated mice that TNF receptor 1 or receptor 2 was knocked out only from neurons. In that point, I want to add that through my PhD, I got over my mouse phobia, so eventually we became friends. Anyway, so in these mice, we induced 2 experimental models of multiple sclerosis, the experimental autoimmune encephalomyelitis and the Cooper's-induced demyelination. And together with the in vitro studies that we generated co-cultures of neurons and astrocytes, we found out that TNF receptor 2 does not promote the disease and generally has neuroprotective role, whereas the TNF receptor 1 is the bad guy, the joker, that promotes the disease and has a small deleterious effect. So selective inhibition of TNF would be a much safer therapy for inflammatory diseases, including multiple sclerosis. Thank you.
So next PhD will be Annabelle Perrier from IP Lille.
So I'm from the Institut Pasteur of Lille and the Center of Infection and Immunity of Lille. And I'm going to talk to you about coronaviruses. So that's a family of viruses which are very widespread because, as you can see, they can infect many animals, including humans. And among human coronaviruses, two are of specific interest to us because they both emerged in the last 2 decades. So those are SARS-CoV and MERS-CoV, which you may have heard about. And actually, they are both highly epidemic, as they can cause epidemics, highly pathogenic, as they can cause severe pneumonia in humans. Yet, we still have no specific treatment against coronaviruses, no vaccines, and there are also yet many knowledge gaps into the coronavirus cycle. So when I started my PhD in Lille, my focus was actually on the coronavirus cycle and more specifically on the assembly step. And I wanted to know how the viral proteins travel into the cell from the site where they are produced to the site where they are assembled into the new virions. So lots of cloning, microscopy, and coffee later. Microscopy for which a better resolution would have been nice. I was able to identify 2 trafficking signals in one of the viral proteins which are involved in its trafficking to the assembly site. So it is a small step towards a better understanding of the coronavirus life cycle and hence towards treatment and maybe vaccines. Thank you.
And the last PhD of the international network will be Adelina Stoyanova from Institut Stefan Angelov in Bulgaria.
Good afternoon to everyone. I would like to thank the organizer for opportunity to be here today. I'm from Institute of Microbiology, the Stefan Angelov Institute of Microbiology. And my work is associated with an approach which aim is to the limitation of the development of drug-resistant from Coxsackie B viruses. The treatment scheme that we used consists of consecutive alternating administration of several anti-antiviral drugs, and the main results we observed decrease mortality rate, increase of mean survival time, and preserved and even increased of viral drug susceptibility after the application of the CAA course. And I would like to also thank I would like to express my gratitude to my supervisor, Professor Angel Gălbuf, and my colleagues for their support and contribution to this work. Thank you.